Recall that the plasma membrane was described as a selectively permeable barrier. So this means, or this implies, that some substances can easily cross while others are prohibited from crossing, or at least have a much more difficult time crossing the plasma membrane. So let's turn our attention to how substances are able to cross the plasma membrane and how they go about doing it. So there are two general mechanisms that substances cross by. There is passive transport and there is active transport. So passive transport implies that substances can cross the membrane without any energy input from the cell, while active transport typically requires that the cell invests cellular energy, which is usually ATP, in order to move substances across the membrane. So let's briefly compare these two, and then we can kind of unpack some of these terms later on. So passive here on the left of this table requires no energy input from the cell, and it is driven entirely by an energy that's called kinetic energy. So the cell does not impart kinetic energy in any way to the substances that are moving. Instead, kinetic energy is simply the energy that is that the moving molecules have at the molecular level. So that is at the molecular level, molecules are just kind of intrinsically moving, and that movement is the kinetic energy of that molecule. So in passive transport, substances tend to move from areas of high concentration to areas of low concentration. So this is referred to as moving down or with a concentration gradient. And we're going to look at this term here in just a second, so we'll unpack that here in a minute. A good analogy for this movement from high to low concentration is water. Kind of picture water moving down a hill with the force of gravity from a location higher on the hill to a location lower down on the hill. So we're moving from high to low. On the other hand, active transport requires energy input from the cell, usually in the form of ATP energy. So we're called that ATP is the cellular energy currency that is constructed by the cell. So with this energy input, the cell has the ability to move those substances against the concentration gradient. So this means that a substance will move from an area where that substance is low in concentration to an area where that substance is in higher concentration. So a good analogy for active transport is water moving up a hill, right? So water is only able to move up hills, or it can only do so if energy is used to move that water. So imagine that energy has to be used in something like a water pump that's going to pump and move that water up the hill against the force of gravity. So there are some pretty simple analogies for passive and active transport, as well as some comparisons and some contrasts to those two types of transport systems. So let's focus on passive transport here for a moment. So we see that there are two passive transport mechanisms. We see, or I should say, two categories of passive transport mechanisms. There is filtration and there is diffusion. So let's look at filtration again for a second. We discussed this a little bit earlier, and filtration is the process by which a substance is pushed across a membrane by some kind of pressure or force on the walls of that membrane. So a good example of this is the production of the interstitial or the tissue fluid from the blood. So our blood pressure pushes fluid out from out of the thin walls of the body's capillaries, and that fluid enters the space between the cells of that particular tissue. And so it's the pressure of blood that is forcing fluid out of the blood and into the tissue space. So that's filtration. We see this in a few places in the body. So we see it in all major capillary beds of the body. We also see it going on in places like the kidney, and we'll discuss that when we get to the chapters in A&P2 that describe renal and kidney function. So our other transport mechanism here is diffusion. And so diffusion is described as the random motion of molecules from an area of high concentration of that molecule to an area of low concentration of that molecule. So in this kind of simple diagram that you see here, the substances that are dissolved in our fluid are referred to as solutes. And you can see that on the left side of this diagram, there is a high concentration of these solutes, and towards the right side, there is a lower concentration of those solutes. And so when the solute diffuses, it will tend to move randomly from this area of high concentration on the left to this area of low concentration on the right. So as I had mentioned earlier, the energy that drives this diffusion is referred to as kinetic energy. And so this diffusion is not driven by any type of energy that the cell gives this solute molecule or anything like that, but it's driven by kinetic energy. And so kinetic energy is not made by the cell. And so as I mentioned earlier, all substances have a kinetic energy, that is the energy of motion, kind of intrinsic to them, and that kinetic energy causes those molecules to move at the molecular level. And so this random movement, this random motion of molecules at the molecular level results in collisions between these moving molecules or particles. And these particles are then dispersed randomly because of those collisions. So we see that kinetic energy is the energy of movement. Because molecules move, there are collisions between molecules, and those collisions cause the molecules to move randomly within that fluid, or within even air as well. So diffusion does occur in air as well. So in this example here of kinetic energy, we have a beaker in which a pellet of dye has been placed at the bottom. And the beaker is, I will just assume, is filled with water here, so we can just assume it's filled with water here. So this dye pellet that is placed at the bottom of the beaker of water will dissolve because of the random collisions of the water molecules with the dye pellet. And so the dye, after some time, will then be dispersed throughout the beaker, even if the water is not stirred, because at the molecular level, the water is moving and it's colliding with our dye pellet. And so it's going to dissolve that dye pellet, and those dye molecules are going to diffuse randomly throughout that small volume of water within that beaker there. So there are actually several factors that influence diffusion rate of substances across the plasma membrane. So when we're talking about rate here, remember that diffusion rate is the amount of diffusion that is occurring over a certain period of time. So anytime we have a rate, we're dealing with something over time. So diffusion rate is simply the amount of material that is moved across a membrane in a given amount of time here. One of the factors that determines diffusion rate is temperature. And so temperature ends up altering the kinetic energy of the molecules that are diffusing. And what we end up seeing is that heat actually increases the kinetic energy of moving molecules. So molecules that are warmer or environments that are warmer tend to have increased kinetic energy, whereas cooler environments or molecules that are cooler tend to have lower kinetic energy. And so therefore, an increase in temperature causes an increase in diffusion rate, while a decrease in temperature will cause a decrease in diffusion rate. So we've probably all experienced this at one point or another with a glass of sweet tea here. So with tea with some sugar in it, right? So if our tea is ice cold and you try to add more sugar to it, and you begin stirring that sugar into your tea with a spoon, we notice that it takes a long time for the sugar to dissolve and diffuse within that cold tea. But if the tea is warm or hot, the sugar dissolves and diffuses more quickly throughout that tea. So that's kind of a real simple experiment that you can refer back to just so you can see how temperature influences the rate of diffusion. Now, when we're talking about dissolving with the sugar there, we're also talking about diffusion there, because the sugar crystals that you put in the tea have to be dissolved, and then that sugar has to diffuse into the tea that you're drinking there. So what else determines diffusion rate? So it turns out that molecule size, so the size of the molecules that are moving, also influences how quickly or how slowly diffusion will occur. So it turns out that molecule size and diffusion rate are inversely proportional, which means that small molecules tend to diffuse much faster than one another, while large molecules tend to diffuse more slowly than smaller molecules. So here is a simple example here with two dyes that have been dropped onto a petri dish of agar, and agar is a water-based gel that you typically would put in a plastic or glass petri dish in order to grow bacteria or something like that and perform experiments in microbiology. But agar also lets us test diffusion as well. So on the left, what we see here is a drop of an orange dye that's called potassium permanganate. And it has been placed on the left side of this petri dish at some point earlier during this experiment. What you're seeing is the end of the experiment right here. So potassium permanganate has a molecular weight of 158. And this term MW, or molecular weight, is a measure of molecule size. So a smaller molecular weight means a smaller molecule. On the other hand, on the right side of this dish, a drop of a blue dye called methylene blue has been added, or has been placed to that on that agar. And methylene blue has a molecular weight of 320. So this means that methylene blue at the molecular level is about twice the size of potassium permanganate. And so notice here that the potassium permanganate, what you're looking at, like I said, is the end of the experiment here. And so you notice that this potassium permanganate spot on the plate here is much larger, which means that this potassium permanganate diffused much more quickly over time than this little spot of methylene blue did. And this is because potassium permanganate is a smaller molecule than methylene blue, and so therefore it was able to diffuse and spread out through this gel much more quickly. Another factor that determines diffusion rate is a factor that's called the steepness of the concentration gradient. So this one takes a little bit of an explanation and an example that I've set up here. So let's imagine on the left side here that we've set up a small container with two partitions, and we'll call them environment 1 on the left here and environment 2 on the right. And between these two environments, we'll put an oxygen permeable membrane, and this oxygen permeable membrane simulates a simple plasma membrane. And then we'll carry out two scenarios, or two experiments, right? So in this first experiment or scenario, we'll add a little bit of oxygen to the left side to environment 1. So we'll add a low concentration of oxygen to the left, and we'll measure the diffusion rate across this oxygen permeable membrane over a certain amount of time. In the second scenario, we'll take the same container that we've set up, and on the left, instead of adding a little bit of oxygen, we'll add a lot of oxygen. So that is, we're going to add a high concentration of oxygen, which is represented by these red dots here on the left side. And then we'll measure the rate of diffusion across this membrane. And so what we end up seeing is that in this second scenario in which in the second scenario in which we started with a higher concentration of oxygen, we would have a higher rate of diffusion than in the first scenario. And so this means that the more stuff that we start with on one side of the membrane, the greater the diffusion rate is going to be. And so if we wanted to look at this in terms of what happens when we increase concentration gradient. So when we increase concentration gradient on one side of the membrane, we're going to increase the diffusion rate across that membrane. So we're going to get more stuff diffusing per unit time. And if we decrease the concentration gradient, that is, if we start with a lower concentration of stuff on one side of that membrane, we're going to get a lower rate of diffusion across that membrane in the same amount of time. So that is hopefully a simple explanation of the steepness of the concentration gradient. Another thing that determines diffusion rate is surface area. And so surface area plays a huge role in diffusion rate. And so if a membrane has a high surface area, or high SA here, the diffusion rate will also occur at a high rate. Whereas a lower surface area will result in a lower rate of diffusion. And this is why we see microvilli in places where diffusion and absorption are particularly important. So in the kidneys and in the intestines, we tend to have cells that line the inner parts of those structures that have millions of microvilli on them. And this is because those microvilli increase the surface area of the plasma membrane of the cells that are carrying out that diffusion and absorption. So more surface area, better rates of diffusion; lower surface area, lower rates of diffusion. And last but not least here, we'll take a look at a factor that is the number of transport proteins in the plasma membrane. Now, some types of diffusion, and we'll get into this in kind of the next segment here with lecture, is that some types of diffusion require transport proteins or protein channels within the membrane in order for that substance to cross the plasma membrane. And the relationship here between the number of transport proteins and the rate of diffusion is directly proportional. So that means that if a membrane has a high number of transport proteins for a particular substance, that's going to mean that there will be a higher diffusion rate of that substance across the membrane. Where the reverse is true when we lower the number of transport proteins, or we have fewer transport proteins or protein channels. So the lower number of transport proteins, or the fewer protein channels that we have in that membrane, the lower the rate of diffusion will be.